Today we'll be studying about cell cycle. I'll give you a brief introduction about cell cycle and the important keywords that are relevant in learning this topic. The first thing that we need to know is about the cell. You know about basic constituents of the cell, the nucleus, the cytoplasm, and other organelles. Now we need to study about how they divide, because without cell division, a cell cannot produce daughter cells, and in that way, the organism cannot survive and it cannot produce newer generations. So, cell cycle is an important process that is going on in our bodies on a day-to-day basis, and in some organisms in minutes. So, the first thing that we need to understand is about these important words. The first one is haploid. We will hear these terms in your course a lot many times. So, a haploid cell is a cell in which only one set of chromosome is present. That is N. That is the number of the sets of chromosome. In diploid, only in diploid, two sets of chromosomes are present. Then these are the terms that I'll be calling when the cell division is taking place. Two types of cell divisions are there. One is mitosis and one is meiosis. So mitosis is where the number of sets of chromosomes remains the same. That is, if a diploid cell is producing daughter cells, then the number of the set of chromosomes that is present in both of them will be 2n. That is, it is maintained. Okay, the set of the chromosomes, number of set of chromosomes is maintained in mitosis, but it is not the case in meiosis. So, can a haploid cell undergo meiosis? No. It is only a diploid cell that can undergo meiosis. That will uh, make the make its condition from 2n to n. That is, from carrying two sets of chromosomes, it will carry only one set of chromosome. Okay, that is mitosis and meiosis. So, which parts of our body, which cells of our body undergo mitosis? The somatic cells. That is, all other cells except the gametes. The gametes are reproductive cells. Okay, so gametes are produced by meiosis, or there are some cells there which will undergo meiosis and produce gametes in our reproductive organs. Okay, so uh, keeping these terms in our minds, we'll now study about the cell cycle. Cell cycle is a genetically controlled series of changes. Okay, this is a keyword we'll be learning here, genetically controlled. We'll see about its implications later on. Series of changes. That is, a cell does not remain in the same phase all throughout the day, okay, or at every point of time. It is going to undergo some changes. So, that is the series of changes we're talking about. In a newly formed cell, what is the fate of a cell? Let me ask you philosophically, what is the fate of a cell? See, a daughter cell has been produced. What will it go now, undergo now? What will happen to it? Okay, so it depends on how much time it has to do its work. The fate of a cell is simple. It gets produced, that is, a daughter cell is produced. It will now start preparing for getting divided into a number of cells, two cells, in fact. Okay, so in a newly formed cell, helping it in genome duplication. So, cell cycle is helping, matlab, what is happening in the cell cycle is genome duplication, the synthesis of its constituents, the cellular constituents, other constituents, growth of the cell, which is also important, and because we need to have enough growth so that the two daughter cells that are going to be produced get enough of it, like equal division of the constituents has to happen for the two cells to be healthy. So for that we need to have growth in the cell to give it to the two daughter cells, and that will result into finally division into daughter cells. ठीक है. So what is genetically controlled? This is an important term which has great implications for other important topics that we will be studying in biology and in future when you go into a medical college, or if you want to understand about serious health issues, for example cancer. You need to understand the genetic control that we will be discussing here. A cell is, when it is moving on, like from one phase to another, we have many phases that we'll be studying about that. So when it is moving from one phase to another, there is a strict control by certain proteins and certain proteins which are further under control of the genetic constituent of our body, our genetic constitution. So that is the genetic control that is being talked about here. Okay, so our genetic constituent, मतलब constitution, strives to function in a way that controls the different controls the going of a cell from one phase to another, okay, by employing some proteins or some other uh, protein dependent kinases and other uh, biochemical paths. Okay, so that is the genetic control we'll be talking, and genome duplication, that is what I told. If we have to contribute genetic and other constituents of the cell to two different daughter cells equally, then there needs to be duplication and growth of the cell to have that. So further, first let me tell you about the different phases of the cell cycle. Okay, this is your interphase. Interphase is the longest part that I'll be getting here. This is the G1, S, G2, and this is the M phase. This entire is the M phase, the entire portion. Now there is a G0 phase also. Suppose a cell is instructed to not reproduce anymore, that is not to divide. It will go into a quiescence stage. Quiescence stage, that is G0. It will go into dormancy. Lie dormant, no division for a certain time, rethinking its possibilities. So, first phase we'll be seeing here is G1. This is the S phase, that is the synthesis phase. Here the constituent synthesis and everything is happening. And then G2. Okay? And this is M, as we talked. Okay, G1 to S. Okay. This M phase is divided into prophase, anaphase, metaphase, and telophase. And then what happens? Cytokinesis, and the division takes place. Okay, so this is a ongoing process in a cell. It happens in a cell, then it happens in its daughter cells. It is continuously happening. So what were the genetic, the biochemical components and everything I was talking about? It is here, cyclins. Okay, Cyclins are the proteins uh, and CDKs that are cyclin dependent protein kinases that are present here in G1 to S, that is here, this here, and G2 to M, that is here. CDKs, various families of CDKs are present here. We need not study into that detail, but still, we have CDKs which are gatekeeping this phase change, that is G1 to S and G2 to M. Why? We shall ask why. Why is it happening? Because something very important is going on here. For okay, let's suppose you have something that you want to double. So it will take energy on your part and time on your part. You don't want to waste it. So suppose something is wrong in the cell and it is uh, in the G1 phase, the cell is in the G1 phase, and we come to know that something is wrong with the cell. Do we want to synthesize it or double it? Why to waste our energy and time in it? If there is some problem, some defect in the cell that needs to be corrected. So the CDKs are the components that are going to find out that if they find out any defect in the cell or anything abnormal in the cell, they will alert the other body, the mechanisms, and take the cell will go then to the correction mechanism. It will not go further to the S phase, because in S we are putting so much of time energy to duplicate the constituents. We don't want abnormal constituents to be duplicated, waste of